Our next presenter is Theresa Wrynn from the Department of Oral Biology. The title of her presentation is Spit Happens, Restoring Salivary Gland Function. Some fun facts about Theresa. She has played ultimate frisbee competitively and went fishing during a pink salmon run in Alaska. That sounds... Did you catch some salmon?
I did,
Nice.
yep. And I got a picture holding it, it's slimy.
Sweet. Sounds like fun. All right, Theresa. Ready, set, pitch.
I want to tell you about a situation. The person you see up on the screen had cancer, and in order to get rid of the cancer, she underwent radiation therapy. Now, that same therapy that helped treat the cancer is the same therapy that has now permanently damaged her salivary glands, the organ that produces your saliva. Damage to your glands is not just limited to this situation, however. It can also be caused by other diseases or natural aging. The person you see next to her lives within your glands and is trying to fix the damage that has happened. But more on him later. I want to go back to your saliva for a second, because it's not something you think about every day until you don't have it anymore. Without saliva to moisten your mouth, you can't eat, talk, or chew properly. Without the protection of saliva, you have painful fissures that form on your tongue and increased infections that happen in your mouth. Life becomes quite uncomfortable, and patients are left to live in constant discomfort because we have no cure. We do not know enough about your salivary glands in order to fully restore their saliva-producing capabilities. However, that is where my research comes in, which is aimed at identifying those important components that maintain both function and structure of your glands. Now, this organ is made out of building blocks called cells, and I want you to imagine that each cell is a mini factory where you have managers and the assembly line. However, there are hundreds of these managers, but I can predict which ones are important by using a fancy computational algorithm, and then I can physically remove them from that factory. Through this process, I've identified a manager named XBP1, which I have spent the past 5 years and a multitude of experiments showing that without this manager, those factories stop working. Cells die, the structure of the gland changes, and most importantly, there's less saliva being produced. My hope is that in the long term, this manager remains a part of the conversation as we look towards long-term therapeutic options for people who suffer from salivary gland dysfunction. Because at the end of the day, these patients, especially the ones who have already battled cancer, do not deserve to spend the rest of their life in discomfort. Thank you.